So now this is where things get interesting, all right? All of the energy sources then go to something called the electron transport chain. We're moving on from the citric acid cycle to something called oxidative phosphorylation, which deals with the electron transport chain and chemiosmosis, all right? So where does that take place? Do you guys remember where the electron transport chain occurs? Right, it takes place in the mitochondrial membrane, right? All of these products here are going to travel, all of these energy products are going to travel here, where we have the electron transport chain, all right? So I'm gonna go ahead and draw my little dotted lines. These guys are all gonna go boop. So off to the electron transport chain we go. Now I am going to go ahead and blow up this image down below this drawing here. Sound good? Okay, zooming in on the electron transport chain in our intermembrane space. So just as a point of reference, I'm going to go ahead and draw out this square that we're zooming in on. And then what we have is, right here is our mitochondrial membrane that we're dealing with, where all of our complexes that are a part of the electron transport chain are found. But just for point of reference, the whole structure of all of this can be confusing to some students. I just want to take a second and zoom out a bit. Just as like a brief reminder, here's a mitochondria. And now the mitochondria has an outer membrane like this. So this space in here, all that, that's the outer mitochondrial membrane. And then inside of the outer mitochondrial membrane, we have something that looks like this. And so this whole thing, that's the inner mitochondrial membrane. And so the space that's in between the outer membrane and the inner membrane, this whole area here that goes all the way around, all of this blue, that is the intermembrane space. Remember, inter means between. Sorry, I ran out of room there. But inter means between. So this is the space between the outer inner membrane. So this is the intermembrane space, okay? So over here in our square, all right, this is a mitochondrial membrane. Specifically, this is the inner mitochondrial membrane. And out here, this is the intermembrane space. So down here, this is inside of the mitochondria. So this is what we call the matrix of the mitochondria, or the mitochondrial matrix. And so referring back to our image over here, all of this jazz in here, that is the matrix. It's the inner part of the mitochondria. Sound good? All right, so this is the mitochondrion. Outside of the mitochondrion, we have the cytosol and all the other organelles that are floating around. Make sense? Okay, so now that we have a little bit more orientation, let's go ahead and dive into the electron transport chain, all right? So zooming in, we now have, thanks to the TCA cycle, a bunch of goodies to work with to help us produce some energy, right? So from the citric acid cycle, we were given several electron carriers, such as NADH and FADH. Ooh, can I spell? So those came from the TCA cycle. And now it's time to see how these guys that are each carrying electrons with them, how these guys are then going to donate their electrons to the electron transport chain in the effort to produce energy. So along the electron transport chain, we have several complexes. We have complex number 1, complex number 2, complex number 3. We have a couple of helper guys as well. And then we have complex number 4. And then at the very end here, we have the big bopper. We have ATP synthase. And if it has synth in it, that probably means it's synthesizing something. So yes, this is where ATP is synthesized or made, all right? So the goal is to get all of our electrons down the line so we can use them to help produce ATP, or energy for the cell. So let's break down how this happens, starting with NADH. NADH carries 2 electrons, all right? And it donates those electrons to complex number 1. And this process produces NAD+ as well as a proton. As the electrons move through this protein complex, energy is gradually released, and this energy is used to pump hydrogen ions, or protons, that are inside of the mitochondrial matrix into the intermembrane space. The electrons are then transported by this special little molecule here towards their next destination, to complex number 2. And it's here that electrons are actually donated by FADH2, which produces FAD as well as 2 protons, because it's FADH2. And then all of these electrons are then passed on to another protein complex. At this point, this next protein complex, the electron energy is used to once again transport hydrogen ions across the membrane and into the inner membrane space. So you might have noticed then that suddenly we have a pretty darn high concentration of protons or hydrogen ions in this intermembrane space. This will be very important soon, so keep this in the back of your mind that we are increasing the concentration of protons in the intermembrane space, all right? That concentration is becoming pretty darn high as electrons are being passed down this process. Now the electrons are transported to a final protein complex, where their energy is used to once again pump hydrogen ions into the intermembrane space, making the concentration even higher. Now the electrons need somewhere to go, a final destination to complete their journey. And so here's where oxygen comes in, right? You often hear of oxygen as the final electron acceptor, and this is why. So this is why oxygen is so important and why it's a necessary part of this specific process. Oxygen, not technically O2. So oxygen at this point will come in and accept electrons as well as some hydrogens. So oxygen comes in and accepts the electrons as well as hydrogens from the matrix, making water. This is one of the main reasons why your cells need oxygen, to accept these electrons, or the electron transport chain will grind to a halt. Now it's time to touch on the last part of the process where we actually get to synthesize some ATP. So remember that the concentration of hydrogen ions we've been building up in the intermembrane space? Well, now it's time to use it to make ATP, a protein called ATP synthase that we have over here. That's my cat again. So here we have ATP synthase. This makes a channel that allows hydrogen ions to pass through from high concentration of hydrogen ions to a lower concentration of hydrogen ions. Protons, like everything, naturally want to travel down their concentration gradient from an area of high concentration to an area of low concentration. Remember going through that earlier in the semester? Yes, so same rules apply. So this is a process, remember, known as diffusion. But since they have to travel through ATP synthase to get back into the mitochondrial matrix, we would call this facilitated diffusion, the passing of those protons through a channel into the mitochondrial matrix. So the facilitated diffusion of these hydrogen ions through ATP synthase actually causes the ATP synthase to spin like a turbine and produce, from this process, ATP. So since we made these since we got these many electron carriers from the citric acid cycle, right, cellular respiration, the electron transport chain happens over and over again. So from this process, we ultimately produce anywhere between 30 to 36 ATP molecules in total. And so if it helps you to think more about this mathematically speaking, you can think about it this way in terms of the electron carriers and their equivalents to how much ATP they produce in the electron transport chain. So roughly for every one NADH molecule, you get about 3 ATP. For every FADH molecule, you get about 2 ATP. And then for every GTP molecule, you get about 1 ATP. And then obviously 1 ATP is equivalent to 1 ATP. So if we want to scroll up and count everything up to make sure it all makes sense, over here from 1 pyruvate, we got 4 NADH, 1 GTP, and 1 FADH. So that would mean that for 2 pyruvate, so for 1 glucose molecule, you end up with 2 pyruvate. So if we wanted to look at this from the perspective of a single glucose molecule, we would actually have 8 NADH, 2 GTP, and 2 FADH. And so that would mean that we would essentially have 8 times 3 ATP from the electron transport chain, which would be 24 molecules of ATP. For GTP, 2 times 1 is 2. Hello, Smudge. That's the blind cat. He's okay. He's sitting right here with me. And then we have for FADH, 2 times 2 is 4. And so adding this all together, we end up with 30 ATP. But remember, this is just from the citric acid cycle. We also need to add in the energy that we are going to get from glycolysis, right? So zooming out a bit further, from glycolysis, we got 2 ATP and 2 NADH. So from this, we get 2 ATP, as well as 2 times 3 for NADH to ATP, and that would be 6. 2 plus 6 is 8. Now, I know what you're thinking. 30, last we checked, plus 8 is equal to 38, not 30 to 36, right? Well, this is because what we are looking at here is in a perfect system where no energy is actually used to help facilitate the movement of electron carriers, right? So 2 things to keep in mind are, number 1, the system we are looking at is within a living system, right? This is within living cells. And things that happen in life in general don't happen flawlessly, right? Nothing is 100% energy efficient. The other thing to keep in mind is that to make energy, we tend to need to spend energy. A perfect example of this is something that we see in glycolysis, right? We see that we have an energy requiring phase where we used up 2 ATP just to get this whole process started. And the same holds true if we want to transfer molecules like NADH and FADH, right? If we want to transport these electron carriers to the electron transport chain for oxidative phosphorylation, that actually costs energy. And on average, it usually costs around 2 ATP to transfer these molecules. But it can take more because remember, like I said, this is not a flawless system to bring these molecules to where they need to be. So this is why whenever we study the production of ATP through cellular respiration, we provide a range of ATP we would expect to get after this whole process has concluded. Make sense? So on that note, I want to actually now synthesize this in a way that hopefully helps people keep all these numbers and procedures in mind, okay? So over here, I'm gonna go ahead and erase 30 plus 8 is 38, because I think you know that. So keeping in mind that if this were a perfect system and we got maximum ATP yield, we would get 38 ATP from the system. However, that's not very realistic. Now over here, I'm going to draw out a table for us, and we're gonna fill in this table together to summarize everything that we've gone over in this procedure of glycolysis and the Krebs cycle and oxidative phosphorylation, including the electron transport chain, together so far.